Hi everyone, welcome and good evening. My name is Jasmine, and on behalf of Harvard Bookstore, the Harvard University Division of Science, and the Harvard Library, I'm excited to introduce this virtual event with Matthew Cobb, presenting his new book, As Gods, A Moral History of the Genetic Age, joined in conversation by Kevin Davies. Thanks so much for joining us virtually this evening. Tonight's event is the latest installment in our Harvard Science Book Talk series, which works to bring the authors of recently published science literature to our Cambridge community and beyond. To learn more about this series and our other upcoming events, you can visit harvard.com and sign up for an email newsletter, or check out the page harvard.com/science for more info. This evening's discussion will conclude with some time for your questions. If you have a question for our authors at any time during the talk tonight, click on the Q&A button at the bottom of the screen and we'll get through as many as time allows. In the chat, I'll be posting a link to purchase As Gods on harvard.com. Your purchases and financial contributions make events like tonight's possible and help ensure the future of a landmark independent bookstore. Thank you to our partners at Harvard University, and thank you all for showing up and tuning in support of authors, publishers, indie bookselling, and of course, for science. Now it's my pleasure to introduce today's speakers. Matthew Cobb is a professor in the School of Biological Sciences at the University of Manchester. He is the author of 6 very popular science books: The Idea of the Brain, Life's Greatest Secret, Generation, The Resistance, 11 Days in August, and Smell, a very short introduction. Tonight, he'll be joined in conversation by author and journal editor Kevin Davies. His most recent book is Editing Humanity, The CRISPR Revolution and the New Era of Genome Editing, and he is the founding editor of Nature Genetics, as well as the executive editor of the CRISPR Journal. This evening, they've joined us for a discussion of Matthew Cobb's new book, As Gods, Moral History of the Genetic Age. This book traces the history of genetic engineering, showing the importance and dangers of this revolutionary technology. Deborah Bloom of the New York Times raves that Matthew Cobb illuminates his search for decency and honor in a morally complex field in As Gods, as he dreams of a world in which the public is better educated on the issues, in which scientists engage with the larger community, and informed regulation is possible, though in the complicated reality that technology has built for us, far too often, dreams and nightmares must go hand in hand. We've got a lot to learn this evening, so without further ado, I'm delighted to turn things over to our speakers. The digital podium is all yours, Matthew and Kevin.
Okay, thank you very much, and good evening, everybody. It's 11 o'clock over here in the UK, but I will not cease from giving you my talk. So I'm going to share my screen, and there we go. So we are actually just gone past the 50th anniversary of the development of genetic engineering. It began in the autumn of 1972 when Paul Berg, who's at Stanford University, published this little article in the Proceedings of the National Academy of Sciences. And although humans have been altering genes ever since we appeared on the planet, because we're predators, because we will alter the genes of the animals and the plants that we eat, this was the first time that we have been able to deliberately and precisely change a genome. And Berg's work actually led to a scientific revolution. Within 5-6 years, researchers from Genentech were able to develop a new way of producing insulin. They used genetic engineering to introduce the components of a human insulin gene into a bacterium, which was then literally within months after this picture was taken, they were able to produce it commercially. And this led to an utter transformation in our production of some drugs. Any of you who take insulin today will take it from use genetically engineered insulin. And this also led to the explosion of the biotech industry, vast wealth for some people who joined the right companies at the right time and took out the right patents. But that's continued. I mean, Insulin is the big success story, but that's continued right down to the present day. I'll just give you one example. This paper that came out only a few months ago. And unlike the Genentech researchers who did something very, very clever, but relatively simple, this research involved 56 different genetic edits in yeast, including introducing 34 genes from plants in order to enable this yeast to produce this, or the precursor of this new cancer drug vinblastine, which we can produce by more traditional chemical means, but this is much, much more efficient. So this is a process of using genetic engineering to produce new drugs, which is ongoing and which is really transformed the way that the pharmaceutical industry works. But as most of you will probably know, the big change has really happened in the last 10 years, when just over 10 years ago, Jennifer Doudna and Emmanuel Charpentier announced that they had invented CRISPR gene editing, and they were eventually awarded the Nobel Prize for this, Nobel Prize for Chemistry in 2020. And the point about CRISPR is that it is absolutely extraordinary in its simplicity and its cheapness. It works in virtually every organism. It's very, very simple to do, relatively speaking, and it is much, much cheaper than the alternatives. In terms of therapy, or even if you're using it as an experimental tool, which is the main way that CRISPR has been employed, you've got to get the relevant bits to the right tissue in order for it to produce the change you want. So for therapeutic aspects, this has actually been quite difficult. And there are some tissues that we can target, like those in the blood, which are relatively easy to get hold of. Tissues, for example, in the brain are much more difficult. But you can do extraordinary things with CRISPR in an experimental context. So in animals, experimental animals, you can literally turn a gene on at the flick of a switch by using what's called an optogenetic construct. You can put this next to the gene that you're interested in using CRISPR, and you then shine a light on your organism and the gene starts work. I mean, this would have been science fiction. If anybody said, 10 years ago that we could do this, I would have just laughed at them. It seems absolutely bizarre. Very soon after CRISPR was invented, a group carried out a proof of principle. In fact, it was at Harvard. They deleted human genes one by one in a cell line to see how those genes, how the cells would react with or without various genes to a whole load of drugs. This is something that would have been completely impossible without CRISPR. So it's transforming scientific discovery and will transform medicine. Here's one very famous example. This very brave woman is called Victoria Gray. Some of you may have heard of her. She suffers from sickle cell disease, which is a disease which primarily affects African Americans and those of Afro-Caribbean origin, and people in Africa, but not exclusively. So white people can have sickle cell disease as well. But the main thing about sickle cell disease is extremely painful, very, very debilitating. In May 2019, she underwent an experimental treatment. So this isn't a therapy, it's an experiment, very brave. And they used CRISPR to start a form of hemoglobin, which is the molecule that transports our oxygen, to change that from the form that we have as adults, which in Victoria's case is got this strange sickle cell form because of the way that the proteins are organized, to produce fetal hemoglobin, which is a different kind of hemoglobin. Meant basically she's fine. The edited cells is working. She feels much better. She said she was without pain for the first time in her life. So the medics are still very cautious, but this is the kind of thing we can do. We have remarkable possibilities for new treatments. Here's another example: xenotransplantation, the possibility of using kidneys, for example, from pigs to overcome our lack of kidneys for kidney transplantation. The big problem is that we have the pig genome has lots and lots of kind of trapped viruses that are inert. Then they don't move when they're in the pig, but if you put them into a human, then these retroviruses start popping about, and that could cause an awful lot of problems. So in 2017, again, this is partly done in Harvard, all of those genes were removed from these rather cute little pigs. And since then, 3 human genes have been introduced into the pig genome so that any kidneys that were to be introduced would have much less likely to cause an immune response. And this is now, there are trials going on with transplants into primates, and we wait to see what's going to happen. So this is another possibility. And finally, a kind of an additional version of CRISPR, rather than editing whole sequences, just changing single bases, which again has been developed at Harvard by Alexis Comore and Nicole Gaudelli, and it was Kevin who took this photo. In fact, all this has been done in the lab of David Liu. And for example, they've recently knocked down a gene that is involved in high cholesterol levels in a monkey, and a single injection of these base editors led to a reduction in cholesterol levels by 60%. So there are the possibilities of huge, not only discoveries, but also applications in medicine. But from the very beginning, what I want to concentrate on for the second half of my introduction is the problems that are associated with this. And I think it's very striking that from the very beginning, the genetic engineering community has had a reaction to the science that it is doing that is unique in the history of science. Even before genetic engineering was a reality, in 1971, Paul Berg was convinced not to do an experiment because it might be dangerous. With the development of what was called gene cloning after Berg's experiment with 1972, the whole scientific community became very concerned that the experiments they were doing could be dangerous. And they called a moratorium, a pause, a research pause, which was global and was obeyed as far as we know. And that is unlike any other science. Even the scientists working on the Manhattan Project, building the atomic bomb, they had their doubts. They wrote a letter to Roosevelt, which mysteriously wasn't sent, but they protested, but they carried on working. Genetics is unique in that respect. And we'll see that this principle of being concerned by what you're doing and the possibilities that it could go wrong and taking steps to avoid that are actually still present in the genetic engineering community. I'm going to concentrate on 3 new possibilities and potential threats very, very briefly. Firstly, I'm going to talk about pathogen manipulation. I'm going to start by saying that there is no evidence that COVID-19 was genetically engineered. Okay, there is a tiny little possibility that maybe it was being reared in a lab and it escaped inadvertently, but all the evidence at the moment we have points to a natural spillover event, probably from some kind of bat. But I don't want to talk about that. I want to talk about the research that is being done and that is dangerous. So throughout this century, there have been a series of events that have alarmed researchers. In the year 2000, a group of Australian conservationists accidentally made mousepox vaccine resistant. Now, mousepox is, as the name indicates, it infects mice. Now, you might think, well, why is that worrying? Because mousepox is incredibly closely related to smallpox. And the procedure which they carried out, which made the mousepox vaccine resistant, if the same principle were applied to smallpox, then all of us who've had smallpox injections would now be, have been vaccinated and are immune to the disease, would now be susceptible. And all our vaccines that we've stockpiled in case smallpox were to reappear would be useless. So this was extremely alarming. After 9/11, there was great concern about the possibility of bioweapons being used by rogue states or terrorists. And this led to a massive expansion of US research on pathogens and trying to predict how they could be made more dangerous. First, researchers reconstructed the 1918 flu virus, which is far more dangerous than COVID-19. And we know that the West's warnings that this is really dangerous and the terrorists could get hold of this technology actually drew the interest of Al-Qaeda. After the fall of Afghanistan, US troops discovered documents written by the deputy leader of Al-Qaeda saying, the West says this technology is very dangerous, we will try to apply it. Now, as far as we know, they weren't able to do so, but they wanted to. the outbreak of SARS, which is another coronavirus, which appeared at the beginning of the century and was basically, we dodged a bullet then, we were very lucky. Basic public health measures in China and around the world stopped it from spreading, stopped it from getting out, like we've had with COVID-19. But the outbreak of SARS led researchers, in particular in the US, but around the world, to try and think, well, what could possibly go wrong? How bad could this get? And this was the development, therefore, of gain of function studies. You've probably all heard of this. That is researchers finding a disease and trying to see what would happen if it mutated further. In other words, they're trying to make the disease worse in order to be able to predict what might happen. So these aren't crazy people. They might be ill-advised, but they're not crazy, and they're doing it for the right reasons. But in 2012, we had another crisis moment, because in 2012, Ron Fouchier, who was a researcher working on gain of function studies of bird flu, H5N1. H5N1 is very, very dangerous indeed. There's currently an outbreak of it around the world in birds, but it's not a problem for humans unless you touch an infected bird. So that's the reason why if you find a sick bird, do not pick it up because you might get ill. But what Ron Fouchier did as part of this project to try and work out what could happen, how bad could a bird flu outbreak be, he did what he called he said it was really, really stupid. He made H5N1 transmissible through the air like COVID-19. And this, frankly, scared the willies out of him and of his colleagues who he explained it to. And so they had another of these research pauses. This time they were expecting it only to last for 60 days. In fact, it lasted for about 8 months. And there was big debate about the funding of such research. Was it a good idea? And so on. Various biosecurity problems in China and in the US with various lab leaks of other viruses led to the questioning again of the usefulness of gain of function research. And this is still debated within the scientific community, but it is going on. Second thing that, again, you probably know about is the possibility of heritable genome editing, so changing genes that will be passed down the generation. This was announced in 2018 when He Jiankui, a Chinese scientist, announced the birth of 2 girls that had been edited as embryos. We now know there were 3. There was another baby that was born shortly afterwards. You got to remember these babies were normal. The embryos were normal before he got his hands on them. They were deliberately mutated. And above all, the procedure didn't work. The wrong mutations were introduced. They're not in every cell. The girls are mosaic, which means they've got a mixture of genotypes in their cells. We don't know what will happen. Hopefully they'll be okay. The Chinese government, rightly, I think, is not turning them into a circus, but this is clearly very, very alarming. As a result of that, a whole set of researchers, Eric Lander, Françoise Baylis, Feng Zhang, and Emmanuel Charpentier, and Paul Berg, who's still around, they called for a moratorium, another research pause on heritable genome editing. But very strikingly, you'll notice that Jennifer Doudna's name is not here. There is not, in this case, unanimity on in the scientific community. So there is no moratorium, and some researchers may be very foolishly continuing with this work. Final thing, perhaps the scariest one for last, environmental manipulation. So the starting point is 600,000 people die every year from malaria. The vast majority of them are children under 5. So that's our target. That's what we're living with at the moment, or those people, those communities are dying with. That's what we've got to try and overcome. And researchers have developed a form of gene that is called a gene drive that can very, very rapidly change entire populations. It doubles its frequency every generation. So it's incredibly effective. And you could conceivably, and you can in the laboratory, either make a whole population go extinct, or you could make them, say, become sterile, no longer able to transmit malaria or whatever. Now, the researcher who came up with this, Kevin Esvelt, who was a student at MIT at the time, said, yeah, I was pretty elated on the first day, thinking this is the answer to malaria and to so many other ecological problems. And then the next day I woke up and I was absolutely terrified. So I actually didn't tell anyone, even my supervisor, for over a month. Now, when Kevin thought of it, it was just an idea, but very shortly afterwards, researchers on the other side of the country actually created one of these things, and they called it a mutagenic chain reaction. It's a genetic bomb. And they were so worried that they worried about whether they should actually publish, because if one of these things gets out, you can't get it back. And Kevin has since spent a lot of time trying to think about ways, clever ways of fixing the genetics of one of these things that you can get the benefits and will lead to it petering out or dying out very, very quickly. But we're not there yet. So I think the message of all this, as Spider-Man discovered, is that with great power, there must also come great responsibility. But the key point, I think, and the lesson of genetics attempts to grapple with this itself is that it is too important to be left to the scientists. And that, as Jasmine said in her introduction, is one of the things that I'm very concerned about, that we need to ensure that the public is as widely informed as possible about the benefits and the dangers of this new technology, because it is extraordinary. And if we are going to apply some of these techniques, we need to understand as a global community what the risks are. And with that, I hope I'm going to talk to Kevin. Okay, I'll stop screening. Stop share. There we go.
Thank you very much, Matthew. And hello, everyone. I'm Kevin Davis in Washington, D.C. A fantastic. Oh, I wasn't expecting
That's
a
his
plug
book.
there. Anyway, this is the book we're supposed to be talking about tonight. So this is As Gods from Basic Books. And Matthew, I'm going to spend the next, hopefully, 25, 30 minutes peppering you with questions around the book and perhaps a couple of other tangential issues as well that I know you're very interested in. You have a whole range of fascinating interests. And then most importantly, I think we want to get to the audience questions. So if you're in the audience and you'd like to ask Matthew a question on just about anything in this book or anything that you think he'd be helpful on, he's here for the full hour. So please put your questions in the Q&A box and we'll try to reserve the last 10, 15 minutes of our discussion to get into that. So Matthew, I know the book is As Gods here in the United States, but it's not the title, I think not your working title. Tell us a little bit about how it comes to have two such wildly, maybe not wildly, but quite different sort of images in across the pond.
Well, I can't remember who it was. Was it Oscar Wilde or somebody said that the United States and England were a common culture separated by to the same language or something like that. But I mean, that's the basic, that's the issue. The US publishers felt that the UK title was a bit ponderous. It's just called The Genetic Age, Our Perilous Quest to Edit Life. And they wanted something a bit more oomphy. So we had a bit of an argument. They won, and they're probably right. I think this goes down better with the US audience. It's got more of a zippy feel to it.
I feel your pain, because 20 years ago when I wrote my book about the Human Genome Project, my working title was The Sequence, which I loved. The Matrix had just come out.
Oh yeah.
And my American editor, our first call was, sorry, Kevin, that's way too abstract for an American audience. It's never going to fly. We're going to have to dumb it down, I'm afraid. And so they came up with Cracking the Genome, which I loathed at the time. But anyway, that's all water under the bridge now. Also, the subtitle is interesting. I wonder if you can tell us a bit about that. It's a moral history of the genetic age. What were you trying to get across with that?
I wasn't. And it's not a word I feel terribly comfortable with. And again, let's just say there was a difference of opinion about this. But I think what the publishers want to get over is that this isn't just a history, and that I have opinions, which I do, right? I mean, I think it's, I write history sometimes, and in this case, I put a bit more of myself, and because it's actually, in a way, it's about politics, about where we're going, what we're going to do. It's about policy, it's about regulation, which makes it sound kind of dry. But if you just think back to the three things that I've just talked about, they're all rather concerning, or they should be. And in particular, when I mean, I've been talking about this to the public since about 2014, something like that, made radio programs for the BBC and so on. And in particular, I would talk back in the day about what would happen when somebody eventually carried out germline gene editing. They changed the genes of a human being. And I would say rather coldly to my students or to the public, well, the worst thing that would happen would be that the baby would die and whatever, and But when it was actually announced, when it actually happened, and He Jiankui, and it was all over Twitter, and we were watching live streams of his talk, I was absolutely raging. It was a big surprise to me. I wasn't expecting that I would be so cross and so furious at what had happened, and so worried for the girls, which I still am. So I think that was quite interesting. So it's partly that it's a book that tells history, and it's got various jokes in it, but it's also got a view. It has an opinion which the reader doesn't have to agree with, but that's a bit more interesting than having something kind of sterile, I think, or simple or academic.
Definitely not that. What was the most enjoyable part of writing this book? I mean, it's largely a 50-year window from the early 70s. You do talk in the first couple of chapters about events before that, but you're really covering the evolution of genetic engineering from the early 70s through to the present day, and then of course projecting beyond that. And much of that sort of ground, particularly as a professor of zoology, would have been broadly familiar to you before you really got stuck into this. So I'm curious what was most enjoyable and perhaps what was particularly surprising as you did your research.
Well, the two bits that really excited me and surprised me for finding them exciting were, firstly, I mentioned it very briefly in my introduction, the Genentech period. So, I mean, you've got to remember, Genentech was founded, I noticed this, I was quite proud of this little factoid, was founded, or incorporated, whatever they do, the same week that Apple Computer was, just down the road in Silicon Valley. So, I mean, this is part of the same world that we now live in, but was completely not even embryonic. It was a glimmer in mum's eye at this stage, with what it might turn out to be. And reading about the early years of Genentech, about how Swanson and Boyer set it up, and then the genius idea of going for of producing insulin, and the excitement of it, the race there was, and then the vast wealth that Herb Boyer ended up with as a multimillionaire when it went public in 1980, the same day that Paul Berg won his Nobel Prize for the invention of the whole thing. Berg never made a penny out of it, I don't think, directly. That was really, really exciting. And I'm not an enterprise kind of guy, I'm afraid. I'm a bit of a European old school socialist. And so reading about this, the real, people putting their livelihoods, their houses on the line, and then producing it, working. Of course, I didn't go much into the people who lost everything because it was crap. That was fantastic. It was really exciting. The other thing that I was very interested in was the original development of GM crops, and in particular, the work of Monsanto, who get a lot of bad press, rightly so, but their starting point was they wanted to develop GM crops to get out of chemicals. And I thought that was really interesting. And I made a little, during lockdown when I wrote the book, I also made a 3-part BBC radio series about this, which is available on the BBC website. Anybody can listen to it. And I talked to people from Monsanto about it. And that was really, really interesting, again, to hear how they'd done it, and the pressures and the excitement and the whole community there was of plant geneticists creating something which, whatever you think about it, has actually changed agriculture in key parts of the world, and above all has reduced the amount of insecticide that we're spraying about.
I'm sure we're going to come back to that theme. Matthew, about a year or maybe 2 years ago, for the 40th anniversary of Genetic Engineering and Biotechnology News, a publication with which I'm quite familiar, you wrote a little blurb for us to celebrate the anniversary, and you said, "Our ingenuity must be deployed not only to discover new potential therapies," which you touched on, but also to find ways of rendering them simple, safe, and cheap. How do you think we're doing in that regard?
Well, there's two ways. I mean, for a start, we could contrast the dreams of insulin, which is as cheap as chips to produce, and yet we know that in the US in particular, it's amazingly expensive. So you can have a great idea, but then there's a whole infrastructure and ecosystem of finance that can cause terrible effects. I think there is a major issue. So there was Eric Topol on Twitter tweeted a set of recent developments in, this is an article, I'm not sure where it is, maybe it's in your journal, about the recent gene therapies that are being developed, and they're all absolutely incredibly expensive. They're very, very specific in general, because they've got to target genes that we're really confident are involved in a particular form of cancer or a particular genetic disease. But they are amazingly expensive. And I think it was David Liu who said at one of the meetings about gene editing, said, we live in a world in which there is not equal access to spectacles. So how can this technology really help health equity and all the problems that we know that exist around the world? And you can think about that with sickle cell disease, because we are going to end up with a very simple cure, I'm convinced, for sickle cell disease, which affects millions of people around the planet. But will that be equally distributed? Probably not. So there are other bigger issues, and this relates to regulation, in particular for gene drives example. And this is where the science comes up against the politics. And your mileage may differ, so the readers can have different answers, but you need to recognize the problems before you can come up with an answer and work out, is this the answer that we as a global community want to apply?
Yeah, as you said in your little summary, Matthew, the CRISPR itself is an incredibly cheap and easy form of gene editing. That's really why it's swept around the world over the last decade. But by the time you've manufactured it and developed the viruses and the vectors, and then figured out the cost of not taking that potential drug and then pricing your drug accordingly, it gets very expensive very quickly.
And I think these are real issues. I mean, again, this is not something, I mean, I'm a professor of zoology. I know about, jellyfish and centipedes and things like that. So I don't know about getting a drug to market, except I know it's really, really hard.
Yeah.
And getting some insight into the commercial world, which is, of course, if you're producing a therapy, then that's what you need to do. It's great to have, it may work in the lab or in a rat, but then actually making it work in humans, changing lives, that is an incredibly long process, and most drugs fail. And it's very, very expensive. And I think these are things that we need to think very hard about how we want to guide this research and encourage it to produce the best outcome for most people.
I wanted to ask about the public perception of genetic engineering, which hopefully is something your book is going to inform in a major way. As you discuss in the book, the past 50 years of genetic engineering has been really, every step of the way, there's been some sort of public outcry or fear about the use of technology, and you go through them all pretty much. There's the early potential misuses, cloning, Dolly the sheep, genetically modified organisms, now designer babies in the wake of the CRISPR babies scandal. Is that does that come with the territory, or do you see that going away anytime soon?
Well, in a way, I think it's going away, which is rather alarming. So, I mean, for example, CRISPR babies, I mean, we all went nuts. The scientific community went berserk, which was very gratifying in a way. It's that, a handful of people initially on the first day said, oh, it wasn't so bad, and then very quickly backtracked. And, so the scientific community was as one that this was awful. Even the people who haven't signed Jennifer Doudna may not have signed the moratorium, but she thinks it was awful. Let's be absolutely clear.
Yes.
She just thinks we can't control it, and so it'll go underground, and you're better off knowing what's going on. So I think I'm not sure that people around the world were really that alarmed by it. And then I noticed this because I was interested in one of the themes of the book is looking at how cultures treated this. So I asked on Twitter, it's my source of all information. I'll be using Chat GPT next time, but this time it's Twitter. What cultural products, films, books, or whatever, have there been this century about genetic engineering? And there really there've been a few books written, you know, but there's been nothing that has grabbed us. I mean, the last thing that really got hold of us was Jurassic Park, right, in 1992. There's been no film about GMOs. It's kind of odd. You'd thought, you know, given the worries there were, especially at the turn of the century, that we'd be making things, but it's very strange. And I think that indicates that public concerns, I mean, in particular in the US, for example, over GM crops, you don't necessarily know you're eating GM food, except if you're eating corn chips, then there most definitely are GM. But I think it's kind of going away a bit. And one of the things I was worried about is this acceptance of some things I'm not worried about GM crops particularly at all, in fact, but these new technologies which could produce great benefits, but can also, they're things we should be worried about. And I want people to be concerned about. I guess somebody's making a techno thriller about gain of function studies, I guess.
Yes, probably.
But you've got to remember, so, you know, Jurassic Park has carried on, but the moral dilemma, which is at the heart of the first film, where Jeff Goldblum's character says your scientists were so busy thinking about whether they didn't think whether they should, that issue disappeared after the first film. After that, it just became, hey, when are the dinosaurs going to get out?
Yes.
You know, what's
Way
going to
too
go
long,
wrong?
in fact.
So we've kind of lost that interest. So it's a challenge to creative types. I'm not sure why it's happened.
It's quite extraordinary to see He Jiankui, who you showed his photograph in your presentation. It was released from jail early. He was sentenced to 3 years for his actions in the CRISPR babies scandal. And now he's back. He's being rehabilitated. He's got a lab and new funders in Beijing, and he's working on his golf game every day. He's tweeting another slow motion shot of his I
had hoped that he would be more discreet and go and work on Drosophila or something.
Yes. Yeah.
But apparently not. So that's very
he still thinks he can make a contribution, I guess. You mentioned CRISPR, and you talked about a couple of other gene editing, precision gene editing technologies, which I agree with you are very exciting. During the course of writing As Gods, did you come across any other technologies or new areas of genetic engineering applications that perhaps haven't had the hype and the attention that CRISPR has, that you think deserve a plug here?
I'm not sure. What you're obviously getting at something, Kevin.
No, really. I just wondered if there was something that kind of tickled your fancy that you thought.
No, I think the main thing is gene drives. That's the thing that really worries me. So that's why I close on that. The gene drive. I mean, it's really, really hard because you've got, you know, I can't emphasize enough, all of the scientists, and I've interviewed a lot of them, talked to them in various circumstances, they are in all these fields. You know, the worst they can be is like He Jiankui, stupid and arrogant. But in general, the vast majority, 99.99% of them, are well-meaning, well-intentioned, including the people who are doing the gain of function study stuff, which I think we should ban myself. But, you know, that's just my view, but it is my view. But we've got this issue with malaria, which is, and other insect transmitted disease, which is incredibly significant. So maybe this could be a way to solve the issue. Or maybe, and I think this is the point, and we'll get onto the audience questions now in a minute, you've got to remember that we always want, it's interesting, there's this kind of desire for a sexy techno fix. It's what Robert Oppenheimer called the sweet spot of finding the solution, the really exciting solution, without necessarily thinking it through. And for example, with the case of gene drives, there's recently been a WHO trial of malaria vaccine, which was incredibly effective and will reduce malaria transmission if it is rolled out, which they're intending to do. So maybe we don't need that sexy techno fix. Maybe the excitement of gene drives is just something that we can put to one side and say, well, actually, we don't need that because good old vaccination is going to do the trick.
Yeah,
and I think we need to think that through, you know, cheaper, safer, more reliable solutions to the same problem.
We've got an audience question on gene drives, so we'll come to that in just a couple of minutes.
Okay.
Before we get into audience questions, please keep them coming in the Q&A box. Matthew, I just wanted to widen the lens a little bit, because I know one of the books that you wrote before As Gods was Life's Greatest Secret, where you looked in particular at the events leading up to and beyond the discovery of the double helix, which celebrates, if a helix can celebrate its anniversary, it's the 70th or platinum anniversary of the construction of the iconic double helix next month. What are you? I imagine you're working on something. What are you working on to mark that anniversary?
Well, I'm doing two things. In the long term, it won't be finished by April. I'm writing a biography of Francis Crick, and I've just got to the end of 1953. So I know the story. Although you might think that, but then, today I discovered new things in the archives that were very interesting that I'm not going to tell you about, which kind of flesh out the story that we know. And some of that is going to be published in, I hope, in an article in Nature, which is the journal that published the double helix, as well as the supporting evidence from Rosalind Franklin and from Maurice Wilkins. I'm writing it with Nathaniel Comfort, who is a researcher who is writing a biography of Jim Watson. So we are writing about this. I think it's going to be really interesting. We're writing about the same period, and for some of this time, but not for a great many years, they work together, but we're looking at it from literally different points of view. So that's been absolutely brilliant, working with Nathaniel. We spent the summer going through, we went down to Churchill College, Cambridge, and went through Franklin's documents, all her archives, and looked at them together, and helped us to understand what she was trying to do and why she didn't quite get there in the end.
I know you've spoken at great length, and I'm sure she will feature heavily in the book that you're going to be working on, the contributions of Rosalind Franklin, which were dismissed or certainly underplayed for many, many years, thanks in no small part to the way she was portrayed in Watson's book, The Double Helix, which of course inspired
I think actually he made her famous. If Jim hadn't been a git in the way he portrayed her, she wouldn't have the drive. I mean, I was looking today at I was just going through the London Times and the Sunday Times and looking for the frequency of Rosalind Franklin, and she's mentioned kind of 3 times between 1958 when she died and the turn of the century. And then or 1970s, and then it starts to kick off after the publication of the double helix, and it really kicks off in the new century. So I mean, people always say she's the forgotten lady. I mean, she's not forgotten. We've got a coin in the UK with some of her data on it. So I mean, there's a university named after her and buildings named after her, right, left, and central, whatever. But I mean, she's very, very significant, extremely important. And Nathaniel and I have made some significant breakthroughs in understanding, in particular, why how Watson and Crick gradually changed over the course of the year after their discovery, or made explicit exactly what they had done by using her data in particular.
Even though the model, the double helix, was built and relied almost solely on her experimental data, she was, I think it's fair to say, but you're the expert, she was good friends with Crick especially, and Watson, and stayed with him for long periods, even when she was quite ill. Yeah,
she's very, very good friends with Crick, and in particular with Odile Crick, Francis's wife. They got on like a house on fire. Some fantastic letters from Odile describing going to the theatre with Franklin, and Franklin came to parties at their house and danced until 4 a.m, and so on. So they had a whale of a time.
Well, we've got about 15 minutes left, Matthew. Let's turn to some audience questions, and I hope the audience will keep these questions coming in.
I'll try and keep the answers short.
It depends how many more we get. Bill asks, first of all, says, great talk. What are governments, Matthew, doing to monitor possible pathogen development by quote unquote evildoers?
Yeah, well, governments, not so much, but there are a whole series of attempts by researchers, and different initiatives have been looking at, in particular, the sequences, to try and find patterns in the sequences of newly identified pathogens, to try and see. I mean, you may remember unfortunate statements that was made at the beginning of the pandemic was that there was a signature of genetic engineering in the COVID-19 sequence. This isn't the case, but it did cause a great deal of excitement in the beginning of 2020.
Yeah.
So but that is not in principle impossible, and it may well be possible to trace both the development, because we're going to meet new pathogens, they're going to appear. But part of the worry is that this technology is extremely simple. But I think we need to emphasize this: producing a mutation is relatively simple, but actually then if you were an evildoer and you wanted to release it, that is very, very tricky. And weaponizing these things, that information is not published. So scientists do indeed release all the stuff about how they introduced a particular mutation into a virus, into a bacterium. But what they don't do, I'm glad to say, is explain how you can then, put it in the reservoir, or whatever all the fantasies that were in the 70s that were going to happen. So they are, I mean, I hate to say this, but I think in a way we're reliant on the security services. But given that they didn't even know that the Russians had a massive 20-year program of genetic engineering of bioweapons in the 70s and 80s, I'm not sure we can be entirely reliant on them.
Right. Next question is, and I think we talked about gene drives. You talked about gene drives, Matthew, but I don't think we sort of assumed that everyone knew what the concerns were. And somebody wants to ask, I don't understand the biology of the genetic bomb concern. Could you explain briefly?
Okay, so let's imagine you wanted to get rid of mosquitoes from a particular area, because if you're living, say, in a village in Burkina Faso, which is one of the places that they're looking at for this, a lot of your children may be dying. So you can understand why you would be very keen on scientists coming along and saying, right, we're going to get rid of the mosquitoes because we can make them sterile and therefore eradicate them. As I say, this works in laboratory populations. The issue is, what will then happen? What will then happen both locally? So let's say we remove all of a particular species of mosquito. Now, it is the case that none of these mosquitoes are the sole food source of any other animal. So no animal is going to immediately starve to death, right? But ecology is really complicated. And if lots of, they are eaten by everything, because you've got to remember, they're not just those buzzy, annoying flies. They have a larval stage. So loads and loads of fish, other aquatic organisms will eat them at that stage as well. So you've got, they're an important part of the ecosystem, and you remove them. And if some species just go a little bit hungry, that might be untoward. You don't know we don't understand enough about ecology. Secondly, there is, how do you keep them where they're supposed to be? They move. That's how Zika virus got into Latin America. That's why we've got the creeping patterns of the various insect transmitted diseases that are going on around the planet. The mosquitoes are moving, they're changing their location. So a village in Burkina Faso might decide, but they would in fact be deciding for everybody. I mean, what Kevin Esvelt put it at one stage is that a release somewhere is a release everywhere, because this gene, unless we can come up with some clever kind of inbuilt breaks, which Kevin's been thinking about quite hard, that they will just, the gene drive will just expand and go everywhere. Yeah, which is a bit alarming.
Yeah, and you're a zoologist, so you're this is home turf for you, I think.
Kind of, yeah.
Next question. Hi, Matthew. I'm curious. Getting off on a little tangent now. I'm curious what you think about the current rise of AI and the popularity of ChatGPT, which I know you've been playing with. How can we understand what's going on now through the lens of science ethics?
Well, yeah, I mean, ChatGPT is very worrying. If anybody hasn't tried it, I suggest you go and do it and ask it a question. Ask it to write you a thank you letter to your grandmother for buying you a Christmas present, and it will write something really pretty, well, for a start, grammatically perfect, which is one reason that I think educators know that they will be able to pick out if students do use it, because its grammar and its punctuation is brilliant. But this is it's not conscious. I mean, and it doesn't know anything. It's very, very stupid, and it will produce references. If you ask it for a reference list, say on CRISPR, rather than going to the Wikipedia site and just getting all the articles by Dowden and Charpentier and all the rest of them, it will make stuff up. So you get these references that look really sciencey, and then you think, wait a minute, I don't actually know that. And you look for it and it doesn't exist. So they don't understand anything. But I think there is big ethical issues because, I mean, Nature has got, tomorrow morning, has got an editorial coming out about the use of GPT and other LLMs, they're called, in science. And they say for a start, nobody should use them. And if they do use it, if they've used them in their analysis, they've got to explain how they did it. And above all, nobody should be clever and have the AI signing the article, which a number of people have already done. And they make a very good point. They say being the author of a scientific article means you take responsibility for it. And these things, they don't know anything. They literally do not know anything. All they can do is copy patterns. They do it remarkably well. But I think it's educators that are going to have a problem, because if you give short questions that are relatively simple to answer, so up until kind of second year university level, it can do them. And so the students are going to have to learn to handwrite, or we're going to have to come up with clever, sharper questions. But it's going to be an arms race because the AI will get better as well. I don't know what the end point is going to be. It's very worrying.
Right. I've got 3 more questions in front of me from the audience. So if you want to get in the queue, please enter your question quickly before we wrap up. Matthew, what are the 2 or 3 top issues that involve critical consequences about which geneticists are widely divided?
Well, I think gain of function is probably the most debated one, in that it is stoutly defended by many virologists, right? And I'm not a virologist, I'm not an epidemiologist, so I don't, I have no expert knowledge. On the other hand, I have an educated layperson's knowledge, and it scares me, and I'm not convinced by the usefulness of it. And I think that it's so dangerous that we shouldn't be doing it. But there are very strong arguments on the other side. So I think that is the main one that there is debate about. I'm not sure, the gene drives, everybody's worried about them. And as I say, the people who invented it got worried straight away, in this excellent tradition that genetics has had of thinking hard about it. And apart from a handful of people, I don't think that there is now any real appetite for using CRISPR or any form of gene editing to cure genetic diseases in embryos. Yeah. I think the dangers that are associated with that, and the recognition that in fact very few couples could be helped by this, a few hundred around the world, right, is the estimate. I think that's changed people's minds. But
that field has gone very quiet, hasn't it, over the last 4 years?
Who knows what they're up to?
I'm a little worried that suddenly there's going to be a CRISPR clinic in
well, they could
medical
be
tourism zone announced.
You don't need to do tourism. I mean, just remember, in the US, there is no law preventing anybody with private funds from doing what the hell they like.
Right.
So, and this has come as close as damn it in some US private universities. Yeah. So maybe it's happening now. You're absolutely right. Yeah. But at the moment, I think, a lot of the transhumanist nonsense about it's going to live on Mars and, having our genes. I mean, I think people have realized that's just garbage, which it is. I'm sorry for transhumanists in the audience, but I have got no time for it.
Let's close with these couple of questions. Matthew, what's your view on benefit sharing of genetic resources? How can benefits of genetic data from biodiversity, for example, be used to protect biodiversity?
Yeah, I think this is very interesting. And I mean, it relates as well to genomes. I mean, human genomes, which are the most valuable thing. And Iceland famously sold its whole population, 800,000 people, all their genes. And this is I think this is a really important issue. And with biodiversity, I think there's a great concern, and in particular, indigenous peoples around the world are very concerned that either their genes or their their genes, or the genes of the animals and plants and microbes that live with them, could be exploited. And I think that the main way you get over this is by having co-production. You've got to do this with, not only with you going and talking to people, say we want to do this, but actually creating at root level in these countries. You've got to create the resources and the knowledge amongst a new layer of scientists who you can actually then engage with. And for example, this has been done a bit in New Zealand, where they've been engaging the Maori community very, very closely to ensure that these things aren't just, doesn't become a new gold rush, because it could be. One final point is there's biodiversity and biodiversity. There's an article just out pointing out in Current Biology, pointing out that most of the biodiversity that we all think about involves big hairy things, or maybe birds, and the insects and a lot of the other stuff, which is actually what most of it, the variability depends on, that we tend not to think about.
I'm just going to slip in a quick question here, which we kind of touched on. One of the frontiers of genetic engineering at the moment is de-extinction, and there are projects underway to try to quote unquote resurrect the woolly mammoth, which is quite well intentioned, I think it's fair to say.
Oh,
but
yeah.
A little speculative. I'm curious, you're a zoologist. What's your thought on this endeavor?
Well, an intemperate tweet back in 2015, when this first came up, an intemperate tweet on my part led to me being a world expert on it, which I'm not at all. And I was on the British TV, and then journalists were phoning me up because I said this was a very bad idea. Now, I think the group Massive, as it's called, which is now one of the startups that's doing this, have rode back on their initial ambitions, and it's not de-extincting. They're going to kind of make an Asian elephant a bit hairier and more cold adapted. I mean, okay, that's nowhere near as bad as de-extincting an animal which I really don't think you can do. I know George Church, he emailed me. If you're listening, George, I'm sorry, I know I've got to reply to you, explaining that they could do it all, but we don't even know how many chromosomes these animals had. But maybe that's not an issue. I just think, there are good things being done with genetic engineering cloning. So the black-footed ferret is they're increasing the genetic diversity by cloning a dead animal, and that's good. But I'm not convinced that, much as I'd be very happy to see a mammoth, don't get me wrong, it'd be great. I don't think it's really the best use of resources, but perhaps it's not a zero-sum game. That would be their argument, that we're actually attracting money from the Kardashians or whoever, who aren't going to pay to preserve some slug, right? But they might give a load of money to this research. So
let's close with
mealy
one final
mouth, get
question.
out.
Yes, let's close with one final question from the audience. Matthew, due to the ethical concerns with genetic research, what role, if any, do you think governments and international institutions should play in this research? Well,
that's the big problem, is that, yes, I think this has to, certainly the human genome, the three areas I'm talking about, genome editing, the gain of function research, and especially gene drives, need international regulation. We currently have international regulation of two very dangerous technologies, one of which everybody listening and watching will have done, and that's civil aviation, right? And that's the only reason you can fly safely the vast majority of times, is because of this international organization, which is voluntary. You don't actually have to sign up to it, but nobody will get on your plane or land at your airport. So that on the one hand, and the other hand, the International Atomic Energy Authority. But both those organizations were set up after the Second World War, when the world had an appetite for regulation. And we're not in that same world. The US isn't even part of the International Criminal Court. So I don't think that there's the appetite in the US in particular for signing up to such an agreement. So there are bigger issues in a way that we have to resolve. But there are models for this. There are these two organizations which exist at the moment that regulate on international agreements two vital technologies which can be very dangerous. And I think that's the way to go, but it's going to be a long haul.
Well, Matthew, I think the clock is about to strike midnight over there in the UK, so we should probably let you go. The book is As Gods, A Moral History of the Genetic Age. The link is in the chat, and it's available from Harvard Bookstore and all good independent booksellers. You can find it in other titles in other countries. And Matthew, it's been a thrill for me talking to you over the last hour. Thanks so much, and best of luck with the book.
Thanks a lot, Kevin. It's been great. Thank you very much to the bookstore for putting this on, and thank you much to everybody for watching.
Great. Thank you so much, both Kevin and Matthew, for being here with us. Thank you, Matthew, for staying up so late to talk about this issue that's so thorny and ever thought-provoking and definitely isn't going away. We definitely appreciate your time. And on behalf of Harvard Bookstore, the Harvard Division of Science, and the Harvard Library, all here in Cambridge, Massachusetts, thank you everyone for joining us here tonight. Have a great night.
Okay, bye-bye.